Thank you, everybody, for joining us for our first event after ISAA 2025. I'm seeing some familiar names in the attendee list, so thank you for joining us again. Hopefully, had safe travels back from Brazil. We had a great symposium. The ISAA 2025 edition had lots of really great feedback. So, if you did attend, make sure you submit the feedback in the email that was sent to you. We had 400 attendees from 30 countries, so that's incredible. And then the next ISAA will take place in 2028 in North America. So, very exciting stuff going on. We hope that you enjoyed the last edition and are looking forward to the next one. And without further ado, we have this wonderful webinar that was put together by the Stepan Agricultural Solutions team. So welcome to this webinar, Understanding Biological Formulation Development, Essential Insights for New and Experienced Formulators. And I will now pass it off to our lovely moderator, Sarah Woollick.
Thank you. Okay, well, hello everyone. My name is Sarah Woollick. I'll be your host today for the webinar on understanding the biological formulation development. I'm the technical director for our North American Agriculture Solutions team here at Stepan, and I'm joined by two of my colleagues for today's webinar. Joao Vilela is our market growth manager for ag. He's got a bachelor's degree in agronomy and more than 20 years of experience in the industry. We're also joined by Renaud Perron, a senior formulation scientist for the ag group at Stepan. Renaud has a master's degree in chemistry and more than 10 years of experience in agriculture formulations. So we will be your team for today. First, I wanted to share our LinkedIn poll results and thank everyone who voted in our pre-webinar poll on LinkedIn to really capture what trends you feel have the biggest impact on farmers today. You can see the results were pretty varied, but right now commodity pricing is clearly seen as having the biggest impact by more than 40% of our respondents. That's really why we're presenting on this topic today. Optimizing your formulation to maximize the value of the components that you're using is so very important to help mitigate the pressure of those commodity prices. We all want formulations that will deliver the best value and best performance, and that's what you're going to hear about in our webinar today. So our plan for today, Joao will kick off the webinar talking about global trends in ag and the importance of biologicals. Then Renaud is going to present on fundamentals of formulation development and some critical parameters of compatibility and shelf life for biologicals and some of the ways that he has found to optimize his formulations. He'll also give us a bit of a case study example to really walk you through how formulating brings all that information together to create a solution that will then be used for either biological or conventional active ingredients. We'll finish the session with a Q&A, so please post your questions as we go through the presentation in that Q&A box, and we'll get through as many as we can at the end. Okay, and with that, I will hand it over to JP.
Well, that's awesome. Thanks a lot, Sarah, for the introduction. Also, Parker. It's a great pleasure. Good morning, good afternoon, good evening, everybody. It's really a great pleasure for me and all the Stepan team after a great event at the ISA that held in Rio de Janeiro to keep talking with all of you. It's a great opportunity for us to share in what we have been doing internally at Stepan and also share some trends thinking about agriculture in the future. So my idea here is just a quick introduction, try to give you some food for thought, some things to think. Our pop star here, it will be Renaud. He has a great know-how, and we are going to see all of this know-how and all of these capabilities on that topic that I believe it will be really important today. So just starting, Sarah already mentioned of a poll that we had at the LinkedIn. So a lot of new trends, global trends and new demands on agriculture that we could see that's really challenging for farmers and in agriculture. So mainly the three blocks that we know that are so important is the increase of water, nutrients and input use efficiency. Then we have about yield, That's so important when we talk about farmers' perspective, and also the sustainability. We know that the demands are increasing year by year when we talk about sustainability. And talking about that, we know that farmers have a lot of complex environmental with all of these aspects that we already cover in the pool. And we are, as a company, Stepan are trying to focus in some areas, try to address all of this complex farmer environmental. So going to the next one, please, Sarah. So a very important, when we talk about sustainability, we have a very important piece. It's, of course, that's not the only piece, but biological is playing a very important piece when we talk about sustainability. So try to make agriculture more sustainable. And we have biologicals in several ways to helping farmers to produce. So we could have biofungicides, bioinsecticides, bionematicides. We could also have biological acting as a plant health. And we know that biologicals have been playing an important role for a while. And the importance is getting more and more in all regions, in all crops. And we mentioned that biologicals alone are getting so far. But when we talk about formulation, it will increase a lot the biologicals. So it's getting more muscle. It's combining biological with improvement on formulation. So we enhance the options for crop protection. Please, Sarah.
Okay, we wanted to give everyone a chance to participate in the webinar today as well. So you'll see a poll pop up there on your screen. And we'd like to hear from you. So what do you see are the biggest challenges with biological formulation development? Got a few options for you here: compatibility, formulation, the storage, the shelf life, technical performance, or all of those. So we'll give everyone a few seconds here to pick your biggest challenge. And then we don't have a drum roll, but if you imagine a drum roll, Parker, can we see the results of our first poll here? What are people saying are the biggest challenge that they have?
So it is a tie right now between shelf life and all of the above.
Oh, okay. Well, you are in the right place, everyone. So thank you for joining us today. This is really what we're here to talk about. With these challenges, a lot of these challenges, all of them, and kind of what can be done. So.
Thanks, Sarah, again. So, and just to finalize all to my part before passing the ball to Renault, as I mentioned, biological is getting better and more efficient year by year. A lot of players, a lot of industry, a lot of academy working on that. And then as we put in the pool, there is still some challenges when we talk about biological formulations that needs to be addressed. And we have been working on that, try to getting more science and more winning solutions for the farmers, because by the end, the most important piece when we talk about that is related to the farmers. So we have the mainly challenges, it's compatibility, formulation, storage, shelf life, and the technical performance. So we are going to cover now the formulation aspect that we consider a very important one. So I'm going to pass the ball to Renault to give your show time. Please, Renault.
Thank you, Jean-Paul, and hello everyone, and thanks to the ISAA for having us today. It's very nice to be able to present our work again after what we presented here in the symposium. Now it's a good occasion to do it live and have some interaction with the customers. I see there's already a question in the chat asking what our definition of biologicals is. So there several different definitions, but here biologicals means live things, live bugs, so living microorganisms. So just a quick overview on our research team. So at Stepan, we have different teams in different sites. We're a global company. We have, I think, 25 production sites, and we also have several R&D sites. So we have, of course, formulation development teams. So with the 6 labs across 5 countries, So that's where we do usually things related more to synthetic active ingredients, but it's been a couple of years now that we've built our expertise also in biologicals. We have an agronomy research team, so we have a new greenhouse that's been built a couple of years ago in the US, in Georgia, where we can do some application testing. So, to be able to generate results directly in-house on plants and really see how the products perform. And we also have the luck of having a biosolutions team. So in-house microbiologists who can help with all sorts of testing, like viability, compatibility, and I'll get to that in more detail in the next slides. Next slide, please. Okay, so the first step when you want to make a formulation for ag is to choose the formulation type. Which type are you going to choose? And usually, if you use a synthetic active ingredient, this is defined more by the physchem of the active. Is it solid at room temperature? Is it water soluble, oil soluble, etc.? Obviously, with a microorganism, it's a little bit different. You can't put it in a solvent. You're going to have different challenges. So there's three main formulation types that are commonly used. So you've got the water-based products. I was going to say problems. You got suspension concentrates, but with those, you're going to have issues with contamination and preservation. Living cells like water, obviously, so they're going to want to develop in the bottle, and you don't want that. Now, you could have solid formulations like water dispersible granules or powders. So those are a bit more practical because you don't have the water, but then you can still have some viability issues. And usually what we've seen on the market is that the performance is really not great. And then you have oil dispersions, which are usually much more efficacious, and also being an oil, you don't have the problem with water of the cells developing, but those are usually a bit more difficult to formulate. And that's why we chose to focus on this formulation type by providing you with a solution that's already already optimized for you. So the first step when you work with microorganisms is to look at compatibility. So the coformulants that you will put into the bottle, they might very well just inhibit or even kill the microorganism, which of course you don't want. So the first thing we did was to use this paper disc method, which is commonly used in hospitals to test the sensitivity of bacteria to antibiotics. So basically, you put the coformulants on paper discs on agar plates, and then they're inoculated with the bacteria or the fungi. You let it grow with the right temperatures, right conditions, and then you look, whether there's inhibition or not. So if you look on the left, there's no visible inhibition around those little paper discs. That means this coformulant was compatible. And to the right, there's quite a large halo around each of those paper discs. So that means this coformulant was, probably not compatible. You probably don't want to use it in your formulation with this microorganism. So the issue with this is that each microorganism has to be tested for compatibility with each of the coformulants you want to use. So that's a lot of work. And for anyone who's worked with Petri dishes, that's quite a lot of work. You need to be in sterile conditions. And at the end of the day, even with all the goodwill of the world, there's only so much space that you have in your lab. And so, a few hundred of these Petri dishes, that starts to take a lot of space very quickly. So at Stepan, we thought, okay, how can we improve on this, and how can we develop a method with a higher throughput and with less use of consumables? Which I will talk about on the next slide, because first we have some results of this particular test. So here is an overview of a few of the products we tested with this test method. So we had a couple of gram-plus bacteria, the very classical Bacillus subtilis, thuringiensis. Gram-negative was Pseudomonas fluorescens. And then different fungi, which are used either as fungicides or as insecticides. And then the last one is Aspergillus brasiliensis. Even though it's not directly used in ag, at the time, since we had it in the lab, it was a good model because it resembles some other fungi. So we could also extrapolate some info from this. So what we can see is a lot of coformulants have a very good compatibility, which is indicated in blue. And then as you change some of the properties of the molecules, you start to see sometimes some inhibition. Usually what you can see is that coformulants that are less irritant, usually that have less hazard phrases on the label and the SDS, are usually milder also to microorganisms. And things that are more things that have like sulfates and phosphates and things like that are usually a bit more harsh to the cells. And that's no big surprise, because in microbiology, you would sometimes use, for example, sodium lauryl sulfate as a compound to lyse the cells. So literally just kill them, because you're going to just emulsify the cell walls of the cells. And obviously, that's not good if you're planning on making a biological formulation. So I'm not going to dwell too much on this. If you want more information, you can get in touch with us. You can have all the data and more details on the strains, the exact strain numbers, and we can talk about which specific information you might need. And so to improve this method and to go faster and have more precision on the data, we used a method using resazurin. So that's this bluish molecule on the left, which is initially, it's bluish in color and weakly fluorescent. And whenever there's something alive, it will get absorbed by the cells, and the cells will reduce the compound. And this will give this more red, pinkish compound, which is also highly fluorescent. So how it works is that we put on these 96 well plates. The cells are put at a certain amount. I think it's 10 to the 6 per milliliter. And then we put gradually smaller amounts of the coformulants. So initially a high concentration, and then every time it's the concentration is divided by 2. And so what we look at is at which concentration you stop having the cells working, the cells really functioning. So you can see on this example picture, if it's blue, it means at this concentration the cells didn't do anything. They didn't really live. They didn't really work. And the first red cell is where something's going on. Some life is happening. And so that means, okay, at this concentration, that's the maximum amount you can put of your coformulant for the cells to still be alive. So the good thing is that with this method, instead of going with 3 concentrations, you test 10 concentrations of each coformulant in a logarithmic scale. And then on the same plate, you can put different microorganisms, which you can do obviously with a Petri dish. And this is also allowing to have a scenario where you're mixing the coformulant and the cells in liquid media, which is closer to what happens in a formulation, especially in a liquid formulation. Because in the Petri dishes, it's all based on diffusion on the agar gel, so it's a bit more hit and miss. So this is closer to real life scenario, and this method allows for 30 times faster faster turnaround. So it's a lot more efficient, and it allowed us to generate a lot of data with greatly reduced space use and waste. And here is some of that data. So here we reported for each microorganism the maximum concentration that showed no inhibition here. So depending on the coformulant, every time there's a maximum concentration that was tested. So here when we report, for example, for the first one, make on TD50, which is a wetting agent, with the maximum tested was 10%, and we saw no inhibition. So that means you could probably put more than 10% and be okay. So here also, this test allowed us to use a few more strains. So we have used here a blend of bacillus spores, and we have the two different things. So we have the spores and the actual cells, and that's interesting because it gave us some slightly different data depending on which one you use. So that may be also a food for thought, whether you want to formulate the actual cells or the spores, because the viability might be affected differently. And yeah, we've started to do a lot. We've generated a lot of data here, and we're still generating more. So again, if you are interested, we can provide you with the data, and we can do testing for more coformulants if you need, and we can test with your own strain if you're interested. So just come and let us know, and we'd be happy to help. So far I've only been talking about biology, but I'm actually a chemist. So let's look at how we can make an actual formulation with all that data. So again, just a reminder, we've chosen to go with oil dispersions, ODs, and here's how to develop one. So what's an OD? Because not everyone in the room is a formulator. So an oil dispersion is basically a stable suspension of active ingredient within a water immiscible fluid, which we usually call an oil, as the carrier, creating a concentrate. This concentrate is diluted into water prior to use, like any other pesticide. It's diluted in the tank by the farmer, and it needs to form a spontaneous oil in water emulsion, and the particles also need to be properly suspended. Here, the biologicals, the cells, and whatever substrate comes with them, that's the particles that need suspending. And here's a poll question.
Another opportunity for a poll question. Okay, ODs, we would like to know, what do you think is the most difficult part of developing an oil dispersion or OD formulation? So we've got a few options here. Is the biggest challenge that you've seen finding that right carrier? Is it getting the right emulsion stability for that dilution? Or is it really maintaining viability of the organisms that you're dispersing with the formulation? Or are you not familiar with OD formulations? So those are the choices that you've got. Give a few seconds to think about it. Pick your most pressing challenge. And Parker, maybe we could get the results and see what that one looks like. Okay. Yes, emulsion stability. A winner there, but not too far behind is maintaining viability. So again, I think those are, finding the right balance of those, is definitely, we understand, can be a challenge. So hopefully you'll see some options and solutions here as we continue the presentation. So thanks everyone for that feedback.
And definitely, I would have answered the same. My main challenge developing this product was emulsion stability. So how does the process go to develop an OD formulation? So the first step is to choose your carrier fluid. So which oil you want to use. You have, of course, less options with microorganisms than with conventional pesticides. Then the big chunk of work is to identify an appropriate emulsifier blend to make this nice spontaneous emulsion whenever you mix with water. Then prepare the mill base, so adding the active ingredient, which it might need grinding, because when you have some things that are over 50 microns or 100 microns, you have no choice. You must mill. At some point, whenever the product is going to go through the spray nozzles, they go through filters first, which are 100 microns in size. Therefore, milling is sort of mandatory. So there might be different methods to mill more or less softly. but it's something that will have to be done at some point. Then you might want to combine with additional ingredients, so rheology modifiers and like antifoams and whatnot. Then, of course, check the dilution in water. So looking at dispersibility, suspensibility, the emulsion stability. So all the things you do usually with any other products to make sure that the performance is here in terms of formulation. And of course, check the temperature stability. So usually you'll do 2 weeks at 54 degrees and a few other temperatures. But of course, with biologicals, you might want to choose other options to make sure you don't fry all the bugs during the storage. So here are a few of the areas where the incompatibility might occur with microorganisms. So obviously, the carrier fluid is the first thing to check for compatibility. You want to make sure that it's super compatible because it will be the bulk of your formula. And of course, the emulsifier blend also has to have things that are mild, that won't just kill your cells. The mill base, again, depending on the grinding conditions, a lot of microorganisms are sensitive, so you're going to have to take good care with this. Also, not letting the temperature go too high, because that might happen during a milling step. The other components, and also at the dilution, if there's of course, things like tap water that contains chlorine might not be great. So there's quite a few things that can go wrong.
And we have one final poll for the group today. So looking specifically at organic formulating, and you'll understand why we're asking about organic formulating as we continue with the last part of the presentation. So no spoilers yet, but we're really interested to know from the group here today, any challenges you may have seen on formulating with to meet organic registrations. I know it varies by country and area, but what have you seen? So are you finding challenges with the ingredient requirements that you're allowed to use, with even finding coformulant options that can match and satisfy organic farming requirements and certifications? Or is it still coming back to that compatibility, the regulatory, the performance of the formulation that you're trying to develop? So we're interested to know, before we get into the last part of our talk today, what you think about organic farming formulating. And Parker, if we could get some answers there. Okay, well, again, yes, compatibility, regulatory, performance. I think tells me we have a lot of formulators on the webinar today. So I'm glad to see that. And hopefully, yes, again, you're going to get some tips and tricks of how to address these things. But clearly, yeah, a lot of help needed in this area, and that's what we're here for. Okay, Renaud.
Okay, so some of the challenges, as mentioned in this poll, it doesn't all come from the actual formulation, but also from some of the requirements you might have for organic farming. So you might want to use, because it's organic, you might want to use OMRI or other regional standards for organic farming, and those, of course, come with some drawbacks. Which will limit the options of coformulants that you have. So finding really suitable coformulants that are working, they're really doing the thing you want them to, and also fit those regulations can be quite challenging. And once you have use this short list, you also have compatibility and other things to look at. So it's working with biologicals is even more challenging than synthetics, and especially on a formulation type like OD, which is already difficult on its own, the whole thing is a real challenge. So you start with a limited toolbox of coformulants. Then you need to find a good emulsion of the oil carrier. And believe me, that takes a lot of time when you're only limited to a few ingredients, especially when some of the most useful, most usual ingredients cannot be used because they're just really so bad for the cells. Then you have to, once you've got a good emulsion, it's not enough. You need also to have good suspension of the active. Then you need to prevent sedimentation, so using a rheology modifier, and then double check again viability, shelf life. So yeah, it's a long iterative process. You need to do a lot of going back and forth, and I would say the development time for an OD, you should look at least 1 year, at the very least, because it's so complicated and difficult. If you want to do an OD in 3 months or 6 months, just nah. It's if you have to do all these steps, it's just really not realistically feasible. So that's why we thought, hey, an all-in-one solution would really simplify that process. What if you had all the ingredients already in one single bottle? Just add your microorganism, do the milling process as is, maybe add a rheology modifier if needed, and then, ta-da, you already got a final formulation. And then developing this formulation in 3 months, that's easy peasy. And that's what I'm going to talk about now. So we started with some compatibility data. So with the zone of inhibition, we had 370 samples on 10 strains. At the time, we only had, with the Resazurin, we had 130 samples on 4 strains, and then we built on that later. So that gave me a toolbox of about 30 surfactants with the one carrier oil that we selected. So initially, the method was to do emulsion tests with the standard CPAC method 36.3, which is the standard one for ECs. And that made a lot of formulation tests in the lab. So the aquariums were full of these 100 milliliter tubes with many samples. So we looked at DOE approach, so design of experiment, to really try and find and optimize the best formulation, the best emulsion possible. So for those who are not familiar with those DOEs, so those are ternary diagrams here. So basically what that is that represents the space of 3 different ingredients. So the black dots are the samples that you actually make. And then you add the data into the software and it does some modeling. And then it models the zones where you have different levels of the parameter you're looking at. So here, those are either the creaming or the oil when you do these emulsion tests. So initially using the emulsion test, that took quite a lot of time. So at some point I switched to laser granulometry because that gives a good compromise. Basically, you look at the emulsion size going through directly putting your emulsion in a laser granulometer. The smaller it is, the better the emulsion is. It's simple as that. And I double checked it with the actual emulsion tests, and it works very well. So all in all, I had about 50 combos of 3 or 4 surfactants where I went through all these sample making and evaluating. So that was more than 500 formulations that were prepared. And finally, many hours later, we had one final formula for which we verified compatibility with 7 microorganisms and assessed performance on 5 different microorganisms. And we called it Step Grow Vivo 101. So here I was thinking of having, I don't know, pictures of like flames and explosions and all that, but we have to keep it to a scientific nature. So here's a picture of the actual sample instead. But no, jokes apart, we've tested the compatibility with it. So here we've tested at a higher amount with this Resazurin test of 33%, and we observed no inhibition with any of the microorganisms tested. So basically, it is very compatible. And again, if you would like to send us your microorganism to test for compatibility, we can definitely do that. And now for some formulation performance, the nitty-gritty, what you all want to see. So with ODs, we test CPAC method 180, which is the dispersion stability of suspo emulsion. So that tests for sedimentation and creaming at the same time. So this was all tested in a medium hardness water of 342 ppm. And here, usually for this test, we look at 30 minutes and 24 hours plus reshake and looking 30 minutes afterwards. So here we had different microorganisms. So we had a Bacillus subtilis, a Bacillus thuringiensis, and a Bovera bassiana. So the first two bacillus, we tested at 35% of active ingredient in the formulation, and that was just so that we could reach the number of about 510 to the 9 CFU per gram. So about the standards that you can find on the market. So here you can see on this first one, the Bacillus subtilis, perfect formulation. At room temperature and after hot storage and after cold storage, there's basically no cream, no oil, no sedimentation. It's really behaving really good. I have to say also that the particle size of all these formulations were about 10, 15 microns with a DO9 of 20 microns. So really the performance you're expecting from an actual synthetic formulation. So same again for Bacillus thuringiensis. The performance is near perfect. And here we have the comparison with a commercial Bacillus thuringiensis OD. And you can see that the results are really not that good. So there's a lot of sedimentation and there's a bit of cream too, which tells us, okay, our product, the Step Grow Vivo 101, is really living, up to the market standards. And then we have Bovera bassiana. This one was a little bit more troublesome. It didn't cooperate as well. I think this comes down to the nature of the substrate that this bug lives on, which was probably a mixture of sugars. So when I used 32% of that, it was not really formulatable. So I had to diminish the amount a little bit. So it's only got 10% of AI, which is still 1/10 to the 9. And here we got much, also pretty nice results. There's a little bit of cream, so 1 milliliter, which is very much acceptable, because usually the values are more like 2 mLs to be acceptable. So still very nice, and no sedimentation. So you still get a very good dispersion of your product. And the thing that needs to be suspended, the cells, the spores, those are really, they're still there and they will be sprayed on the crop. And again, we got a commercial Bovera bassiana OD, which had worse performance. So really, this product provides excellent emulsion and suspension properties for various microorganisms, and we're matching or outperforming commercial formulations. And now a little bit on shelf life, which is something that always comes at some point in a conversation. How does your product do with shelf life? So here we've got data on two different strains. So we've got the Bacillus thuringiensis on the left and the Bacillus subtilis on the right. So we stored the products for up to 26 weeks. And we had two different storage temperatures, one of 25, so room temperature, and one of 45. So to have a sort of accelerated storage. So of course, depending on the strain you're working with, you might want to use different times, different temperatures. But these being spores, they can definitely handle 45 degrees, no problem. And usually in biology, to see if there's a difference between two results, you need to have at least 1 log of difference. And so here you can see that the Bacillus thuringiensis starts at almost 1/10 to the 10. And even after 26 weeks at 25 or 45, we're still above 1E9. So there's less than 1 log reduction, which means no difference, no statistically significant difference. And same with the Bacillus subtilis. So here there's a little bit more variability on the data, but this is pretty usual with living cells because they don't always do what you want. But basically, the takeaway is after 26 weeks, which is 6 months, at 25 degrees or 45 degrees, there is no loss of viability. 6 months at 45 degrees, that's well more than 2 years at room temperature. So that means things are stable. So also, the very nice point about this is this OD chassis is ready if you plan to register it for OMRI. So we are compliant with NOP regulation, and we are planning on registering this with Armory. So that means it's an easy solution, and you are sure that you can put this on the organic market easily and have that nice Armory stamp that the customers know and recognize. So that's really great added value here.
Great, thank you, Renaud. We also wanted to mention before we get into the Q&A portion, and thank you for your questions. Please keep putting the questions into the Q&A. We've had some really good ones come through to discuss in a little bit here. We did want to let you know that if what you've seen today has been helpful, if you still have some questions, if you want more help with your biological formulations, Stepan does offer an advanced formulation services program, which is a department that could help with your formulation work. And really any of the tests or techniques that Renaud has shared today would be available through contract with that part of our organization as well. So in addition to supplying testing and help on your projects or formulation development, the team also does training at our Ag Innovation Center in the United States, about an hour east of Atlanta, Georgia, if you're familiar with the area. So please reach out if there's anything we can help you in more detail on any of your projects or questions or training needs you may have. Okay, and with that, we will get into our Q&A session. So yes, please keep adding questions into that, and we'll get through as many as we can in the time that we have left. So we'll start with the biological formulation questions that we've had come through. One of the questions was, how did you check compatibility with the microbes? Maybe I can go back to that one of those slides if you want to just speak a bit to that compatibility. I can bring that up again. I think it came in. Here we go.
Yes. So maybe you can come back to the previous slide. I suppose it was maybe someone who connected after we covered this bit. So very simply, the microorganisms are put at a certain amount in each of those wells. So I believe it's 10 to the 6 per milliliter. And then you add resazurin, and then you add the coformulant. And each of these wells has different amount of coformulant. So you start with the maximum amount, and then it gets diluted by half, and then by half, and again and again. So you get a logarithmic concentration scale. So if it's blue, it means the resazurin is still as is. It hasn't been absorbed by the cells. And if it's this reddish pink, it means it has been absorbed by the cells and it's been reduced. And whenever there's something alive, resazurin will always get inside and change color if there's life going on. And I apologize to the biologists in the room for not having better words for this, but to simply put, if the cell is doing something, there's a color change. And so you can look at the first red well that you have on this plate, and that's where you get, that's the concentration, the maximum concentration that you could put of your coformulant without having the cells stopping to live. So of course it's not necessarily a direct measurement of exactly how much you can put in your actual formulation, because also the concentration of cells is not exactly the same. But that gives you a very nice first idea to say, okay, this one is compatible everywhere. It's compatible with maybe different microorganisms. Therefore, it's probably more mild and it's probably more well-suited to this formulation that I'm making, for example.
Great. We have a lot of questions about this test. So maybe I'll stay on this slide for a moment and we'll kind of tweak your biological questions here, Renaud. So, one question about this test that came through was, does it measure cell death or loss of viability or growth arrest? If you want to speak to that at all with this or any of the other tests that we have available with our biosolutions and microbiology teams.
That's a very good question. So this method is really a quick method. So the standing time is, I think it's about 24 hours in those wells to really have enough time for the cells to, start doing something. So most I think it will be accurate for most microorganisms. But then beyond that, it's still a screening thing. It doesn't, that doesn't specifically look at things like growth and things like that. Although we might be able to make some more precise testing, for example, with the intensity of the fluorescence, because something more alive, let's put it like this, will fluoresce more. So it's something that could be that could be used, and there are also other chemical indicators that can work. There's another one called luciferin. I think one of our competitors showed it at ISAA, and this is something we also have in mind. So there's different things that are possible, but I would say there is not a one test fits all. You're going to have to look at several tests. At some point, you're still going to want to do the CFU count with a petri dish to make sure that you really have the things that you want. And you're still going to have to make the formulation and store it for so many months at room temperature or more, and really look again if you have still the number of cells that you're expecting.
Yeah, good point. I think there's a host of testing and things that will complement one another to let you know kind of really what's happening within that formulation. So we have things like flow cytometry or just standard enumeration testing that you'd want to run multiple tests, I think, on your key formulations. And when you really get some good, this is a great indicator test to give you a really fast, rapid screen, and you can screen a lot of compounds and combinations of compounds. And then you can really start to build on that with some of the additional microbiology tests to really understand what's happening with that organism in your formulation. Great. And I think there was another question about some of the details of the test. So hopefully we've covered some of that. Just in terms of maybe when there was a question on when the rizazorin is added in this test. Renaud, can you speak at all? Do you know at what stage is it added at the end of the formulation and then the wells are filled?
Honestly, I don't remember. There are some biologists in the room which might be able to answer in the.
Maybe one of our microbiologists can
exactly.
chat.
I think there is one in the room, and my apologies to you if I said anything inaccurate.
Excellent. We'll see if we can get
that I really couldn't tell. But by all means, send us a message afterwards, an email, and we can give you more information.
There was also a question about, is this weight % or by volume for testing coformulants?
One, I'm not sure exactly. My first thought would be it doesn't really matter, but it's probably by volume. But a volume weight with this kind of stuff, it's not going to change by a lot.
Sure. All right. Let's see. I'll just see if we have any others around this test while we're on the topic. I think other ones are getting more into the biological formulations. So,
I can quickly answer that. There was a few questions where people asked, is there a preservative? No, actually, there is not in this product. And that's the beauty of it. If it's an oil, there's no water. You don't need a preservative. That really simplifies things. Also, someone asked if the microbes can grow in this. Honestly, I would be extremely surprised if they did, because there's like less than 0.5% water. So there's not a lot to grow with. So unless you have a super mutant alien microorganism which can just grow an oil without water, but then otherwise there is no growth. Everything stays dormant, and that's also one of the advantages of this kind of product.
Great. We also had some questions with the formulating piece. What milling process would you recommend for biologicals?
So it depends on what you'll have in your lab. So this is, there aren't 30 different methods for milling. It's wet millings with beads. So just industrially, you might want to just use maybe a lower speed or a lower sitting time within the mill to maybe not go for the smallest particle size possible. And, there might be a trade-off between, the performance that you want and the small particle size that you would like to reach as a formulator, and then just keep things under 50 microns, or if possible under 20 microns, but don't let it sit in there for longer than necessary, to make sure that it doesn't compromise the viability.
Great. I think another maybe related question is the process for mixing the biologicals with the Step Grow Vivo 101. If you could maybe speak a little bit to how or where someone should add their biological,
So yeah, you could mix it with any sort of method. So just like standard, impeller in a beaker or something, or industrially in a reactor. You might want to avoid anything like ultra Turax or the really high shear mixing, because that might be a bit too harsh for the cells. But afterwards, yeah, it's the milling step that you'll have to do, and you're just going to want to control the temperature and keep the temperature as low as possible so that the cells don't overheat.
Great. Another question on Vivo 101. Did you test long-term emulsion stability beyond a typical test time? Yeah.
Yeah. So usually, I don't like to wash dishes too much, so they have a tendency to just stay there for a couple of days. And I've seen that most of the formulations, after a couple of hours, what you see is sort of the final result. So even for a day or two, the formulations are still very nicely stable. For the sedimentation, of course, if there's no agitation, if there's no stirring, of course, that settles down. But of course, in a real life scenario, you will always have some stirring in your tank. But really, I don't think you would want to go more than 24 hours. Why would you prepare a solution and keep it for longer than that? But yeah, it's if necessary, it can stay for a day or two, no problem.
Another question about the maximum biological loading that you could put into the Vivo 101 product. Have we found an upper limit?
So I haven't looked too much yet at this sort of like upper limits. I think it all depends on your specific microorganism and what is the substrate it comes on. So I've worked with some things that had like more of an inert substrate, and then that worked fine, more of a mineral substrate. And some of them that come on more like sugary or organic substrates, that can make the whole formulation a bit more difficult. So sometimes you will find with your particular active ingredient a limit in terms of how much you can put in, and then the problem is going to be viscosity, or it's going to be even solid. So that will really depend on your microorganism. So you might have to, at some point, to reduce the loading. And then if you can't reduce the loading, you might have to, and you really want to have that number of cells per milliliter, then what you'll have to do is change your process of the production of the microorganism. Because of course, it's that really limits how much you can put in. So of course, if you have something that's already 1/10 to the 9 on the substrates, then you're not going to go very far. So you want to have something that's concentrated enough in the beginning.
There's a few questions that have come in all around the topic of the efficacy or the kind of field trials or plant trials with this all-in-one formulation. I know there's some things in process. Can you speak at all or give us a preview of what's to come looking at this in efficacy trials?
So currently we don't have efficacy results. The challenge is going to be to find a way and a protocol to have a fair comparison, because we're going to want to maybe compare with a different strain. Let's say we have, for example, the Boveria bassiana, where there is a commercial OD. The problem is, are you sure that the product you're going to test is going to be exactly the same strain, and that on its own, will it have the same performance? Because the problem is, I can't put my hands on the strain of the commercial product because it's produced by some manufacturer. They're not going to supply me like that. So I get what I can get, from various sources. By all means, if you know a good source, a new source, I'm always looking for new ones, let me know. If you want to send me some product to do some testing, let me know. But yeah, so far we don't have biological results. We're looking into it, but so far we don't have a protocol fully set to really look at that.
Stay tuned. More to come for sure.
Sarah, let me just...
please.
Yeah, there's a question related to the farmer's perspectives for biologicals. So I believe it's a very nice question. I have been working with biologicals for a long time, I believe more than 15 years. So it was interesting because the farmer's perspectives changed a lot. So in the beginning, I remember that farmers are always comparing biologicals with chemicals. So in this beginning, the most important things for biologicals, it was performance. The farmers expect to have 95% of performance in control pest and disease, always compare with chemicals, so performance and cost. So nowadays changed a lot. So farmers are considering biologicals as a part of the program. So they are thinking about IPM, integrate pest management, integrate disease management, plant health, seed treatment. So nowadays, for the farmer's perspective, we have been seeing that they are looking for easily and handily products. So this is why we have been talking about formulation, compatibility, on the tank mix compatibility, shelf life. So those stuffs are really important on the farmer's perspective, and we have been talking with several ones to see that. Okay, thanks.
That's great. No, thank you for adding that.
I see a few more questions. So one asked, can we have a sample? Yes, of course you can get. That's the whole point of this talk. We want to send samples everywhere so you can prepare formulations with it. So do send us a message. I think our emails are somewhere in the presentation. Do let us know. You can also just connect on stepan.com and create a profile and request for a sample. We will be happy to supply. And then a few questions about the actual form of the active ingredients that we used. So whether it should be a fermentation juice or more of a solid. It's a lot easier if you can provide a solid form for the cells, because that means there is no water. If you try to use a fermentation juice, then that means you're adding water in the formulation, and of course the cells can keep living and keep germinating, and that might cause some issues. So it's more for solid active ingredients. And there was another one on the rheology modifiers. So at this moment, we would recommend using a silica. I'm not going to advertise for the competitor product, but offline we could of course let you know the actual brand name of the silica used. You just use a hydrophobic silica. Here I've used 2%, but depending on your formulation, you might have to use less or more. And we're also working on our side to develop a rheology modifier package. So stay tuned, and we might have something sometime this year or next year.
Great, thank you. Maybe one last part, a few questions specifically about OMRI. And so I would say when it comes to OMRI certifications, the Vivo does use components that we have seen that would be on the NOP list. And so that would be, compatible with OMRI specifically. If you have any more questions about that, depends on what else you might put in that formulation or what your active ingredient is. But yes, that would be compliant with those certifications, and we can certainly help you, to get your product registered with that. So with that, I think we're at time. So I will thank everyone. A lot of great questions about that compatibility test. So it tells me I think we need to come back with our microbiologists and do a deeper dive on that test. We're very happy with it. I'm very proud of the development that the team has done to create that. So stay tuned for more to come on that in the future. And thank you everyone for joining us today. We really appreciate it. Have a great day.
Thank you, everyone.
Thank you.
Bye-bye.
Thank you, everybody. Bye-bye.